Transcription factor activity ELISA assay kits from ELISA Genie allow you to measure transcription factor binding to DNA in a 96-well ELISA format. Why have ELISA Genie developed over 200 transcription factor activity ELISA assays? Well, there are a number of challenges with current technologies. Current methods of detecting transcription factor DNA binding activity are time-consuming, complicated, and can require the use of harmful radioactive labeling. What's the solution? ELISA Genie transcription factor activity assay kits are a high-throughput ELISA alternative to techniques such as EMSA, CHIP, and others. The benefits are they're faster, easier, and safer to carry out with up to 100-fold greater sensitivity over gel shift assays. What are the key applications for transcription factor activity assays? Firstly, detection of DNA binding by activated transcription factors in nuclear and cell lysates using indirect semi-quantitative ELISA. Secondly, the kits can be used to screen compounds that interfere with DNA binding. Thirdly, establishment of target candidates in drug development. And finally, the kits can be used to understand signal transduction pathways. How do the transcription factor activity ELISAs work? Firstly, each kit comes with a plate pre-coated with specific oligonucleotides. Secondly, addition of cell or nuclear extract containing activated transcription factors is added to each well. That's followed by addition of a primary antibody, which is subsequently followed by addition of a HRP conjugated secondary antibody, and finally a colorimetric substrate, and this is read on a colorimetric absorbent plate reader. What are the key features and benefits associated with transcription factor activity assays? Firstly, there's a very fast procedure. This means significant reduction in runtime compared with other methods such as EMSA, Western blot, etc. Secondly, they're very easy to use. Simple ELISA format means rapid results versus other common methods of detecting active transcription factors. They're also very safe. This eliminates the need for harmful radioactive labeling. And finally, they're high throughput. The 96-well ELISA format allows lots of samples to be run simultaneously compared with other methods such as gel shift assays. Here's a selection of some of the more popular transcription factor activity assays, such as P53, NF-kappaB, P65, c-Jun, and also phosphorylated transcription factors such as phospho-SMAD1, phospho-CREB, and phospho-MYC. Finally, these 200 new transcription factor activity assays overcome demanding challenges that researchers currently face around measuring active transcription factor binding to DNA. So let's work together. Visit ELISAgenie.com to find out more about the transcription factor activity assay kits.